Introduction. Imagine being told in the 1980s that computers would revolutionize every aspect of life, from shopping and dating to the stock market. The idea would have seemed far-fetched, yet it became our reality. Today, we stand at a similar crossroads with genetic engineering. What seems like science fiction might soon be part of our everyday lives. Let's explore the origins of genetic engineering, current advancements, and the groundbreaking CRISPR technology that could change our world forever. Historical background of genetic engineering. For thousands of years, humans have been manipulating life through selective breeding. We've enhanced traits in plants and animals, but our understanding of the underlying mechanisms was limited. This changed with the discovery of DNA, deoxyribonucleic acid, a molecule that dictates the growth, development, and function of all living organisms. DNA consists of 4 nucleotides that form a code, and altering this code can fundamentally change the organism. In the 1960s, scientists began experimenting with genetic modifications. By bombarding plants with radiation, they induced random mutations, hoping for beneficial traits. In the 1970s, scientists inserted DNA snippets into various organisms to study and modify them. This led to the creation of the first genetically modified animal, a mouse in 1974, which paved the way for significant medical and research advancements. The advent of genetic modification. The 1980s saw the commercialization of genetic engineering with the first patent for a microbe engineered to absorb oil. Since then, we've produced crucial substances like insulin and growth hormones through engineered organisms. The first genetically modified food, the Flavor SAVR tomato, was introduced in 1994. Although GM foods have sparked controversy, they represent just one aspect of genetic engineering's impact. In the 1990s, human genetic engineering briefly entered the spotlight with attempts to address maternal infertility by creating embryos with genetic material from 3 individuals. This was a precursor to more advanced developments in genetic modification. Introduction to CRISPR technology. Until recently, gene editing was a complex and expensive process. Enter CRISPR, clustered, regularly interspaced short palindromic repeats. This revolutionary technology has dramatically reduced the cost and time required for genetic engineering, making it accessible to many labs. CRISPR originated from the natural defense mechanisms of bacteria against viruses. Bacteria store fragments of viral DNA in a CRISPR archive. When the virus attacks again, bacteria use this archive to recognize and destroy the invader. Scientists harness this system for precise genetic modifications. CRISPR acts like a programmable GPS for DNA, allowing for accurate and efficient editing of genetic material. The power and potential of CRISPR. CRISPR's precision and affordability have opened new possibilities. It can edit live cells, switch genes on and off, and target specific DNA sequences. The technology is versatile, applicable to microorganisms, plants, animals, and even humans. Notable achievements include using CRISPR to cut the HIV virus from living cells and removing over 50% of the virus from rats. This demonstrates CRISPR's potential to combat diseases like HIV and herpes. Additionally, CRISPR holds promise in cancer treatment by enhancing immune cells to target cancer more effectively. Early trials in the US and China have shown positive results. Addressing genetic diseases. CRISPR's ability to correct genetic mutations offers hope for curing thousands of genetic diseases. Some conditions result from a single incorrect DNA letter. With CRISPR, scientists are developing tools to make precise edits at the genetic level, potentially eradicating these diseases in the future. The ethical and societal implications. While CRISPR presents exciting opportunities, it also raises ethical concerns. One major issue is the possibility of creating designer babies. Genetic modifications in embryos could lead to permanent changes in the human gene pool. Early attempts in China have shown progress, but also highlighted significant challenges in gene editing embryos. The technology could lead to new standards of human genetics, potentially enhancing traits like intelligence, appearance, and longevity. As genetic modification becomes more accepted, ethical debates will intensify, especially regarding its application in reproductive cells. The future of genetic engineering. CRISPR's potential extends beyond individual treatments. It could revolutionize human health by tackling aging, a major cause of death. Some scientists believe we could eventually halt or reverse aging through genetic engineering. Animals with natural immunity to aging might provide insights into this possibility. On a broader scale, genetic modifications could address global challenges, such as improving resistance to diseases or adapting humans for space travel. These advancements could reshape our species and our future. Ethical challenges and global implications. The rise of genetic engineering also presents ethical dilemmas. For instance, what if authoritarian regimes use these technologies for control or warfare? The technology's power necessitates careful regulation and oversight to prevent misuse. Conclusion: genetic engineering, particularly through CRISPR, represents a profound leap forward. While it introduces significant risks and ethical considerations, the potential benefits are immense. We stand at the cusp of a new era where genetic modifications could enhance human health, longevity, and capabilities. As we navigate this rapidly evolving field, it's crucial to balance innovation with ethical responsibility. The future of genetic engineering will undoubtedly be both transformative and challenging, and it's up to us to guide its development thoughtfully.